Good morning, my dear students. I am Bharti Kathuria Sehgal from the SD Vidya School, Ambala, Kant. Last time we discussed about the experiment number 1 to observe stomata in the leaves. Today we'll be discussing the experiment number 2 of biology, which is to study that carbon dioxide is released during respiration. First of all, let's discuss in general what is respiration. We've already discussed about the process of photosynthesis, that it takes place in the chloroplasts, in which water and carbon dioxide are used as raw material in the presence of sunlight to produce glucose and oxygen. All aerobic organisms, including humans and green plants, undergo aerobic respiration, in which the glucose and oxygen are broken down to produce carbon dioxide, water, and energy in the mitochondria. If you all remember, mitochondria, which is called as the powerhouse of the cell, is the site of cellular respiration, where the breakdown of glucose in the presence of oxygen takes place to produce energy, which is stored in the form of ATP. Now let's discuss about the experiment that we are going to do to prove that carbon dioxide is released during respiration. For this experiment, I have taken first of all some germinated seeds. Here I have taken the gram seeds. As you can see, you all must be doing such things in your home by sprouting the seeds of pulses, grams, and others. Now here I have kept it in a vessel, the germinated seeds. One thing I want to tell you here is you have to take freshly germinated seeds, because germinating seeds are the ones which have a high rate of respiration. So this is a very important question, that why do we use germinating seeds only for this experiment? Because germinating seeds have a high rate of respiration. So I am using here the gram seeds. Now, let's discuss what next do we do. Now, I'll be taking a glass beaker, a conical flask, a rubber cork, a small bent tube, potassium hydroxide flakes. Now, in the conical flask, I'm putting the gram seeds, the germinated gram seeds that I told you about. Now, here, after placing the germinated gram seeds, I'll be placing the potassium hydroxide flakes in the small test tube, which I have tied with a thread. Now I'll place it inside the conical flask, cut the thread, and now I'll put some water in the beaker. In this beaker, I'll be putting some dye, because this will help me in the observation of my experiment. I'll put the rubber cork with the bent tube of one side in the conical flask and the other side in the beaker. After some time, I'll observe that the water level in the beaker starts rising. Now, why does this happen? This happens because potassium hydroxide, which I have taken in the conical flask in this small tube, which is hanging here, this potassium hydroxide, it absorbs the carbon dioxide which is present inside this conical flask. Oxygen is consumed during the process of respiration. So here a kind of partial vacuum is created in the conical flask, because oxygen को consume कर लिया seeds ने respiration में, right? कार्बन डाइऑक्साइड जो सीड्स ने लिबरेट किया उसको पोटेशियम हाइड्रोक्साइड ने अब्सॉर्ब कर लिया. इससे क्या हुआ, एक तरह का पार्शियल वैक्यूम क्रिएट हो गया है कॉनिकल फ्लास्क में, जिसकी वजह से ये जो वाटर लेवल है बेंट ट्यूब में धीरे धीरे क्या होना स्टार्ट हो जाएगा, इट विल स्टार्ट राइजिंग, एंड दिस विल प्रूव that carbon dioxide is produced during respiration. That's all for today. Take care. God bless you.